In this session, we will discuss about cells of nervous system. That means cells which are present in the nervous system. Actually, nervous system contains mainly two types of cells: excitable cells and non-excitable cells. Excitable cells are neurons, non-excitable cells are supporting cells. So, excitable cells and non-excitable cells, right? Excitable cells are neurons, that I will tell you after completion of non-excitatory cells or non-excitable cells, right? First, if you take the non-excitable cells, these non-excitable cells further classified into macroglial cells, microglial cells, schwann cells, ependymal cells, and capsular cells. Clear? First, we will take macroglial cells. Macroglial cells further classified into astrocytes, oligodendrocytes. Clear? First we will take astrocytes. Astrocytes are star-shaped cells. Aster, literal meaning of aster is star. So, the cells which are resembles with star. So, those cells what we call star-shaped cells are astrocytes, right? So, these astrocytes further classified into protoplasmic astrocytes and fibrous astrocytes. Clear? If you take protoplasmic astrocytes, as I said, they are star-shaped cells. They contains cell body. From the body, radiating processes, that means cytoplasmic extensions. So, these radiating cytoplasmic extensions are symmetrical. They are symmetrical and thick. At the end of each process, we can found plate-like foot process. These foot processes rest over the capillaries, ependymal cells and pial surfaces of CNS, right? So, very simple, these are star-shaped cells and they contains cytoplasmic extensions. These cytoplasmic extensions are symmetrical and thick. They contains foot process or plate-like process at the end. These foot processes rest over the capillaries, ependymal cells and pial surface of CNS. Okay. Then fibrous astrocytes. These fibrous astrocytes, of course, they are star shaped and they also contains cell body and cytoplasmic extensions, that means processes. These processes are asymmetrical and thin. Asymmetrical and thin. Protoplasmic astrocytes present in the grey matter and fibrous astrocytes present in the white matter. Right? So, this is the structure of protoplasmic and fibrous astrocytes. Clear? Let me tell you difference between these two. Star shaped cells, both. Cytoplasmic extensions are processes. Processes are thick and symmetrical in case of protoplasmic. Here thin and asymmetrical. Right? These are present in the grey matter. These are present in the white matter. Of course, at each end of process we can found foot process or plate-like extension. This plate-like foot process rest over the capillaries, pile surface of CNS and over ependymal lining. Clear? Right. These foot plates, when they are resting over the capillaries or when they are covering the capillaries, it makes one barrier or one gate between blood and brain tissue. Blood and brain tissue. That barrier, what we call blood-brain barrier. Blood-brain barrier. Why there should be blood-brain barrier? Because blood contains some toxic materials. If that toxic materials enters into brain, brain tissue will damage. That's why this barrier selectively permits the material into the brain tissue. That's what this membrane is semi-permeable, or this barrier is semi-permeable. It will not allow all materials which are present in the blood into the brain tissue. Clear? So, this barrier, what we call blood-brain barrier. Clear? Now, we will see structure of blood-brain barrier. Structure of blood-brain barrier. If you take from blood side to brain side, there will be endothelial lining of capillaries. Endothelial lining of capillaries. Endothelial lining of capillaries are continuously lined. There is no gap in between the cells to avoid leakage of blood substance into the brain tissue. Right? And also one more precaution. There will be tight junction between the adjacent cells. Between adjacent cells, here we can find tight junction to prevent leakage. Right? That is one layer of gate or barrier, and second layer is connective tissue. That means basement membrane of capillaries. Next, foot plate or foot process of astrocytes. Plate like foot process of astrocytes. Then here nerve cells will be present. These are nerve cells. Clear? So, this is the barrier or gate between the blood and brain tissue. Right? Then this blood-brain barrier, how scientists found? How did they found? First, they injected one dye through the blood and they observed in the brain tissue, but they couldn't identify any stain of that particular dye or color of that dye. Right? And same dye they have injected through the ventricular system. Through that ventricular system, it has reached to the brain and they have observed that stain in the brain tissue. That means through the ventricular system that dye could enter into it, but through the blood it couldn't enter into brain tissue because there is a gate, there is a barrier, and that barrier is selectively permeable or semi-permeable. That's what this material it couldn't enter into brain tissue. Clear? And here clinically you must be observed if RH compatibility, if there is RH compatibility that leads to fetal jaundice, is it or not? So that fetal jaundice, in that condition bile pigments can easily traverse through this blood brain barrier and damage the basal ganglion of brain tissue. That means what? In case of fetus, that means in case of newborn, this barrier is poorly developed. That barrier is poorly developed. That's what bile pigments can easily cross the blood brain barrier and enters into brain tissue and damages the brain tissue. That condition what we call kernicterus. Okay, we will tell you later, but remember that name, kernicterus. That condition what we call kernicterus. Clear? Then one more example you can see in clinical practice. Take one patient suffering from Parkinsonism, Parkinson's disease. In that Parkinson's disease, patients needs dopamine. Actually, there will be dopamine deficiency. Clear? We should give dopamine. If we give dopamine directly, that dopamine cannot enter into brain tissue because this barrier will not allow to enter into brain tissue. Right? At the time, what you have to do? You have to give L-dopa. L-dopa. Actually, L-dopa is precursor of dopamine. That L-dopa can crosses the blood-brain barrier. Once it reaches to the brain tissue, it will convert into dopamine. Clear? So, that is about astrocytes and the function of astrocytes. Function of astrocytes mainly blood-brain barrier. Clear? Right. Then we will discuss about oligodendrocytes. What is the literal meaning of oligo? You must be aware of this word when you are in the clinical practice. Patient comes and complains that I could void very few amount of urine, but when you are writing that sentences, you will not write same as it is what patient complains. You will write simply oliguria. Is it or not? Oligo means what? Few. Urea means what? Urine. He is voiding very few amount of urine. You will write simply oliguria. Right? So here also oligo means few. Dendro means processes. Cyte means cell. Cell contains few processes. That's it. So cell structure will be like this. This is a cell body with nucleus and which contains very few processes. And what is the function of oligodendrocyte? Oligodendrocytes can produce myelin sheath in case of central nervous system. In the central nervous system, these oligodendrocytes forms myelin sheath. But in case of peripheral nervous system, scan cells will form the myelin sheath. Clear? Okay. Now, That is about oligodendrocyte. With that, we have completed macroglial cells. Macroglial cells. Now, we will take microglial cells. These microglial cells are flattened cells with few thin processes. Flattened cells, and they contain very few thin processes. Clear? Possibly they are migrated circulating monocytes, and they are present in relation with the blood vessels, and they are phagocytic in nature. They are phagocytic in nature. That means if there is any material, that means any foreign material, if it can encounter, it will engulf that. Right? So, microglial cells are flattened cells with very few processes. Possibly they have derived from the circulating monocytes during early postnatal life and terminal part of prenatal life. That means just before the birth and just after the birth, they must be migrated from the blood to the brain tissue. Right? So, that is what they must be present in relation with the capillaries, and they are phagocytic in nature. Clear? Then, squam cells. Squam cells are cells which produces myelin sheath in peripheral nervous system. Anyway, I am going to discuss about myelin sheath formation in this session itself. So, just remember that squam cells will produce myelin sheath in peripheral nervous system. Clear? Then, ependymal cells. Ependymal cells are lining the ventricular system, or ventricles of brain and spinal cord. If you take ventricular system will be like this, and here this is the lateral ventricle, and here third ventricle, this is aqueduct of Sylvius, and this is central canal of spinal cord. Clear? So, this is ventricular system. Lateral ventricle, third ventricle, fourth ventricle, central canal of spinal cord. These structures or these cavities are lined by ependymal cells. They are lined by ependymal cells. Right? And what is the function of ependymal cells? They helps in the transportation of materials between the brain tissue and CSF. Right? So, that is it about ependymal cells. Then, capsular cells. Capsular cells present around the larger neurons. Capsular cells present around the larger neurons. Where larger neurons are present? Larger neurons are present mainly in the dorsal root ganglion. Dorsal root ganglion. In the dorsal root ganglion, cells are larger cells. If you compare with remaining neurons or remaining cells, the cells which are present in the dorsal root ganglion are larger. Right? So, whenever there is larger cells, whenever you found larger cell comparatively at that places, those cells are surrounded by some supporting cells to give nutrition to that larger cell. Why? Usually almost all cells of the body will have similar size, that means more or less equal size. But if the size of the cell increases more, at that time the DNA which is present within the larger cell and smaller cell equal, right? So the DNA which is present in the smaller cell or normal cell, they can produce sufficient directions to get nutrition and to produce proteins. But if you take larger cell, the size of the cell more, the material, the genetic material which is present in the nucleus is similar, that means same as it is with smaller cell. So this DNA or this genetic material cannot give sufficient directions to the larger cell to get nutrition and to produce proteins. That's what it needs, some support from other cells. That's what these cells are surrounded by some supporting cells. These supporting cells what we call capsular cells in case of central nervous system or in case of nervous system. But the same arrangement you can see some other place, where when you are studying embryology in the oogenesis, you must be aware of it. Oogonia converts into, of course, oogonia after dividing, when it reaches around 7 million cells, after that these oogonia converts into primary oocyte without any further cell division, only they enlarge their size and converts into primary oocyte, right? So if you compare with oogonia, oocyte size is more. But what about the nutrition? I already told you, if the size of the cell increases, those cells are surrounded by some other cells to give nutrition to that cells. That's what oocytes are surrounded by follicular cells. These follicular cells gives nutrition to the oocyte. Clear? Here also same arrangement you can find. Here is the dorsal root. In the dorsal root ganglion. Dorsal root ganglion. This dorsal root ganglion contains dorsal root. This dorsal root ganglion contains pseudo unipolar cells. Pseudo unipolar cells. Cells will be like these. Larger cells. They are larger cells. These larger cells are surrounded by what are the cells? Capsular cells or satellite cells. Capsular cells we can also call as satellite cells. Then these satellite cells covered by one more layer of cells. These are fibroblast cells, and these two layers of cells forms capsule. Clear? So that's it about capsular cells. Clear? I hope you understood the supporting cells of nervous system. Clear? Then we will go for excitatory cells or excitable cells, that means neuron. If you take the structure of neuron, neuron contains cell body and neurites. Cell body and neurite. Cell body contains nucleus and other materials. Along with these material, you can find extra Nissel's granules. Nissel's granules. Right? Now I am taking the structure of neuron. This is cell body of neuron. From the cell body there will be protoplasmic extensions. These extensions, what we call neurites. That means neuron contains body and neurites. This is body. Body contains nucleus, Nisils granules, mitochondria, of course remaining all substances or remaining all organelles which are present in the cell. Then neurites, this is cell body or perikaryon or soma. Then neurites. Neurites further classified into dendrites and axon. Dendrites and axon. Clear? Now we will see dendrites first. Dendrites are short. They are short and branched. Short, branching and afferent. Afferent means they carries impulses from periphery towards the center. That means from the periphery towards the cell body, right? And they also contains Nisils granules. Right? So very simple. Dendrites are short processes and they give branches and they contain Nisils granules and they carries impulses from periphery towards the cell body. Right? So that is about dendrites. Then axon. Axon is long. Axon is long and it is efferent. Efferent means it carries impulses from cell body towards the periphery. Right? At the terminal part of this axon, you can find terminal buttons or telodendria or axon terminals. Right? So, this is axon. This is long, carries information from cell body towards periphery. Then this axon surrounded by myelin sheath. This axon surrounded by myelin sheath. Actually, I am taking typical neuron. Of course, some unmyelinated nerve fibers also there, but I am taking typical neuron. Here, this axon is surrounded by myelin sheath. This myelin sheath is formed by squam cells in peripheral nervous system. That's what, after formation of myelin sheath, the cell membrane of squam cells surrounds this myelin sheath. This cell membrane of squam cells, what we call neurilemmal sheath. What is this? Neurilemmal sheath. Because of presence of neurilemmal sheath only, if there is any damage in the peripheral nervous system, they can regenerate. Right? Of course, you can find nucleus here, nucleus of squam cells. Right? So, very simple. If you take cut section of nerve fiber from inside to outside, here axoplasm. Actually, axoplasm is the cytoplasm continues as a axoplasm. So, axoplasm. This axoplasm surrounded by this black color, what I have drawn, that is axolemma. Axolemma. So, now this cylinder, total axolemma surrounding the axoplasm. This cylinder, what we call axon cylinder. Axon cylinder. This axon cylinder surrounded by myelin sheath. Surrounded by myelin sheath. This myelin sheath is formed by squamous cells. That's what this myelin sheath, actually it is made up of fat, lipids. This is made up of lipids. Surrounding the lipid tissue, we can find few amount of cytoplasm of squamous cells and cell membrane of squamous cell. This cell membrane of squamous cell, what we call neurilemmal sheath. Neurilemmal sheath. Right? And one more thing here, if you take the segment of myelin sheath which is formed by single squamous cell, that segment what we call internode. This segment what we call internode. And the junction between two internodes, this is one internode and this is another internode. This junction what we call node of Ranvier. What is it? Node of Ranvier. Node of Ranvier. Right? At the node of Ranvier, the cytoplasmic membrane, that means cell membrane of squamous cells, interdigitate with each other at node of Ranvier. Right? Then one more thing you can find here, within the myelin sheath you can observe obliquely present clefts or incisors. These clefts or incisors what we call cleft of Schmidt, Lanterman, cleft of Schmidt, Lanterman. Right? So, these clefts are helpful for the conduction of metabolites to the depth of the myelin sheath and to the subjacent axon. Right? So, this is about structure of neuron. Clear? Then, what are the different types of neurons which are present in the nervous system? First, if you take, based on the processes, that means number of processes, based on number of processes, neurons divides into unipolar cells, bipolar, pseudo-unipolar, and multipolar. Right? Uni means single. If cell body gives only one process, that type of neurons what we call unipolar neurons or unipolar cells. Then second one, bipolar. If cell gives two processes, those type of cells what we call bipolar cells. Multipolar, this is common type. If it gives so many processes, those type of cells what we call multipolar. Then what about pseudo unipolar? Pseudo means false. It is false appearance. It looks like unipolar, but it is not unipolar. That means after some distance it gives two branches, right? So that's what this is, pseudo unipolar. Okay, let me explain one by one. Unipolar. This is cell. From the cell only one process arises. This type of cells, what we call unipolar cells. Where can you found? That means example, mesencephalic nucleus of fifth cranial nerve, which is present in the midbrain. Clear? Then bipolar cells. Bipolar cells means cell body which contains two processes, right? So this type of cells, what we call bipolar cells. And where can you found? You can found spiral ganglion of inner ear. That means spiral ganglion which is present in the modulus of inner ear. And one more example is vestibular ganglion which is present in the internal acoustic meatus. Clear? So that is about bipolar. Pseudo unipolar. Pseudo unipolar means, see here, first this is cell with one process. This is looking like this, is it or not? But so this is unipolar and this is also looking like unipolar, but after some distance it is giving two branches here. Clear? So this type of neurons what we call pseudo unipolar. This pseudo unipolar, where can you found? You can found in the dorsal root ganglion, DRG, or sensory ganglion or spinal ganglion. Three names for it. Clear? So that is about pseudo unipolar cells. Then multipolar cells. Multipolar means, multi means many, right? Many processes. Example is this one. That means it is most common type. Clear? So that is about different types of neurons based on the processes. Based on processes. But if you take based on the length of the axon, based on length of the axon, it will be divided into Golgi type 1, Golgi type 1 and Golgi type 2. Golgi type 1 neurons are this most common type. That means axon is very long. But in case of Golgi type 2, axon will be shorter. That means either shorter, sometimes it will be absent. That means no axon only. These type of neurons what we call Golgi type 2 neurons. Golgi type 2 neurons. So in the Golgi type 2 neurons there will be one special feature will be there. In that, just take diagram of it. See, this is Golgi type 2 neuron. It is having the processes either short axon or absence of axon. In this type of cells, there will be dynamic polarity. Dynamic polarity means what? If you take the typical neuron, this dendrites carries information, carries impulses from periphery towards the cell body. Axon carries impulses from cell body to the periphery, right? But in case of Golgi type 2 neurons, dynamic polarity means impulses can pass in either direction. They can pass towards the cell and they can pass away from the cell, right? So that is about different types of cells based on the length of the axon. Clear? Then after this we will see what is synapse. What is synapse? Synapse means it is connection or junction between two neurons, what we call synapse. It is junction between two neurons, so that all these neurons will form chain which will conduct the impulses. So take this one cell, and this cell or this neuron carrying some impulses. These impulses are giving to the another neuron at the synapse. At the synapse. Just imagine this is another neuron. Right? So synapses, they make the all neurons as a long chains for the conduction of impulses. Clear? And if you want to say definition, it is a junction between two neurons. Right? Actually, if you want to discuss synapse, it takes one class. It's very lengthy. But for completion of all small basic things regarding nervous system, I am giving the introduction here. That's what I will not go in detail, just I will give basic idea about synapse. Actually, I am taking the data diagram of this and I will enlarge over here. If you take, what is this? This is presynaptic terminal. This is presynaptic terminal. And what is this? This is postsynaptic terminal. This is postsynaptic terminal. This is presynaptic terminal and this is postsynaptic terminal. The gap between the presynaptic terminal and postsynaptic terminal, this gap what we call synaptic cleft. Synaptic cleft. Right? So here in the presynaptic terminal we can find synaptic vesicles. Synaptic vesicles. These synaptic vesicles are filled with neurotransmitters. Why? Actually, these nerve impulses transmitted from one nerve to another nerve by neurotransmitters like acetylcholine, glutamate, dopamine, GABA, right? So, like this, many neurotransmitters are there. Through that neurotransmitters or by that neurotransmitters, these impulses will be transmitted from one nerve to another nerve. Okay? Actually, these vesicles are filled with neurotransmitters. They will bind here and they will release into the synaptic cleft. Right? And here receptors will be there. These receptors will receive and they will carry the same impulses through this nerve. Clear? So that is about synapse. After that, what are the different types of synapses? This is very simple. It's based on the junction between the parts of the neuron. Imagine here what happened. There is a junction between axon and dendrite. Actually, this is most common type of synapse. Axonodendritic. Axonodendritic. If the junction between dendrite and dendrite, dendritodendritic. If there is junction between the dendrite and soma, soma means body, dendritosomatic. If there is junction between the body and axon, axonosomatic. So these are the different types of synapse. Now we will discuss about reflex arc. Reflex arc is involuntary motor response of the body. Involuntary motor response. Actually, motor system is under our control. That means under voluntary control. But some motor activities will occur without our notice. That means without brain notice. That reflexes what we call reflex arc. Right? This reflex arc having the ability of neuronal integrity. Actually, integration will take place in the brain and brain will decide what to do. But in reflex arc, without brain knowledge, some motor activities will be occur. Those motor activities what we call reflex arc. Right? Just I am taking the structure of one spinal nerve, then I will discuss with that. This is anterior median fissure, this is posterior median septum, here is grey matter having anterior horn, posterior horn continues as a posterior root, posterior horn continues as a posterior root. Posterior root contains enlargement called dorsal root ganglion. Then this is going to unite with anterior root. So here anterior root, this anterior root unites with posterior root and forms spinal nerve. Right? Here imagine when you are studying very seriously, one of your friend prick with needle behind your arm. At that time what you will do? You will suddenly withdraw your limb from there. Otherwise you will take out the limb from that site of pricking. Is it or not? Without knowledge of your brain. Right? Suddenly we will remove it. That reflex what we call reflex arc. Right? Actually this reflex arc contains some components. What are those components? Receptors, sensory neuron, motor neuron, then effector organ. Right? This is in case of simple monosynaptic reflex arc. Right? So here is the receptor. Just imagine this is receptor which is present in the site of initiation of impulses. From there sensory root passing through the posterior root, and this is cell which is present in the DRG, dorsal root ganglia. This is peripheral process and this is central process, and it comes forwards and it will synapse with motor neuron which is present in the anterior horn. From here anterior horn cells takes motor impulses and reaches to the target organ. Imagine here is the muscle here. There is contraction of muscle. So this type of arc, what we call monosynaptic, because only one synapse is there. The example for the monosynaptic reflex is tendon jerk. Okay, so imagine with knee hammer, if you tap or if you hit over biceps tendon, without your brain knowledge, biceps will contract, right? So that reflex, what we call reflex arc, and this is the example for stretch reflex. Stretch reflex contains only unisynaptic reflex arc. But if it is withdrawal reflex, what I have given example first, when he prick, you will remove your hand from there. That means you will withdraw your hand from there. That reflex arc made up of two synapses. So that arc, what we call multisynaptic reflex arc. So at the time, here receptor stimulated by pin pricking, then that information or that impulses passes through the dorsal root ganglionic cell, and here in between the sensory neuron and motor neuron, actually this is motor neuron and this is sensory neuron, in between sensory neuron and motor neuron there will be one more neuron. That neuron, what we call interneuron, and we can also call as internuncial neuron. Internuncial neuron, right? So this is multisynaptic. For this multisynaptic, example is withdrawal reflex. Monosynaptic example is stress reflex or tendon jerk. Clear? So with this we completed reflex arc in brief, right? Now we will see myelin sheath formation. Actually, myelin sheath formed by squamous cells in case of peripheral nervous system, oligodendrocytes in case of central nervous system, that we have seen. Now, what is the process? What is the process of formation of myelin sheath? Right? First, if you take axon and squamous cell, this squamous cell approximated to the axon. First step, approximation of axon and squamous cell, right? So when it approximates, how it looks like, this is axon and here is squamous cell. They have approximated with each other. Next, invagination of axon into the squamous cell. Invagination of axon into the squamous cell. So here is a axon and this axon invaginated into the squamous cell like this. See here, invaginated. Then formation of mesaxon. What does it mean? It means you must be aware of mesentery. Is it or not? The fold of peritoneum which is suspending the enteron. Enteron means intestine. The fold of peritoneum suspending the enteron, what we call mesentery. right? Here also, see this is a fold of plasma membrane of squamous cell suspending the axon. So this fold, what we call mesaxon. So mesaxon formation, mesaxon formation. Then this mesaxon spirals around the axon. That means it will winds around the axon. How? This is axon and this is mesaxon. So see this is mesaxon, no? This mesaxon, it is going on winding around the axon. See, it is going on winding around the axon like this. And in between these folds of mesaxon, there will be accumulation of lipids and completes the formation of myelin sheath. Of course, we can put nucleus of squamous cell. Clear? So this is the formation of myelin sheath. Clear? Very simple. Approximation of squamous cell towards the axon, invagination of axon into the squamous cell, formation of mesaxon, spiralization of mesaxon, in between the folds of spiralization of mesaxon, accumulation of lipids, completion of myelin sheath. Right? So like this, myelin sheath will form around the axon. Now this axon, what we call myelinated nerve fiber. Myelinated nerve fiber. Actually, all white matter, it's made up of nerve fibers because these are surrounded by myelin sheath. That's what they are white in appearance. Clear? So this is about myelin sheath formation in peripheral nervous system. Right? Then what about unmyelinated nerve fibers? Unmyelinated nerve fibers, all these things will not happen. Only nerve fibers invaginate into the squamous cell cytoplasm and there is no formation of mesaxon and there is no spiralization. Just see here, this is squamous cell. Here only one axon, but here many axons invaginated into the squamous cell. Like this. So these are invaginated into it and forms only neurolemmal sheath around it. Only neurolemmal sheath around these axons, but there is no myelin sheath. So these type of nerve fibers, what we call unmyelinated nerve fibers. These are unmyelinated nerve fibers. Clear? Then how myelination will happen in the central nervous system? In case of central nervous system, I already told you, oligodendrocytes will be there. Oligodendrocytes. These oligodendrocytes having few processes. Right? Clear? So this is cell body. Right? Now each process will wind around one axon. See here, this is one axon and this is another axon, this is another axon. These processes winds around these axons and forms myelin sheath, right? So that is about formation of myelin sheath in central nervous system and also peripheral nervous system, right? Then we will discuss about what is tract, what is nerve, what is ganglion and what is nuclei, right? So here, if there is collection of cell bodies, see here, these are, imagine these are neuronal cell bodies, these are neuronal cell bodies. These neuronal cell bodies are collected at one place. These collection what we call ganglion in case of peripheral nervous system and nuclei in case of central nervous system. Of course, grey matter also collection of nerve cell bodies only or neuronal cell bodies. Collection of neuronal cell bodies present in the peripheral nervous system is ganglion and collection of neuronal cell bodies present in the central nervous system is nuclei or grey matter. And these cells will have processes, is it, or axons or nerve fibers. These cells having nerve fibers, no? Right? See here. Collection of axons present in the peripheral nervous system is nerve. Collection of axons present in the central nervous system is tract. That's what we are calling ascending tract, descending tract. They are collection of nerve fibers only, but they are present in the central nervous system. That's what we are calling as tracts. And we can also call as sometimes lemniscus. Those are also collection of nerve fibers only. Now we will discuss about nerve degeneration and regeneration. If there is injury to the nerve in the peripheral nervous system, what are the changes which occur in the degeneration and what are the changes which occur in the regeneration, right? That we will discuss now. Actually, if there is injury to the nerve, there will be some changes which occur in the cell body and some changes which occur in the proximal to the injury, some changes which occur in the distal part of the injury or distal to the injury, right? So we will discuss one by one. First, injury has been occur at this level. Here injured, this nerve has been injured here. When it injured, within 48 hours, some changes which occur in the cell body. Those changes what we call chromatolysis. Right? So, chromatolysis means degenerative changes which occur in the cell body because of injury. So, what are the changes? We will see one by one. First, Nissls granules disappears because liberation of acid phosphatase. From the lysosomes, right? These acid phosphatases, they digest ribose nucleoproteins. So that's what Nissls granules will disappear. Very simple. within the cell body Nissel's granules are there. Those Nissel's granules are degenerated and disappear because of liberation of acid phosphatase from the lysosomes. This acid phosphatase will digest the ribose nucleoproteins. That's what these Nissel's granules disappear. Right? Once these Nissel's granules disappear, one more change occurs in the cell body. Because of increased osmotic tension, there will be indrawing of water. When the water enter into it, what will happen? Cell become enlarged. That means swelling of cell. That means cell undergoes swelling. So cell swollen. Right? Then nucleus is there here in the center. This nucleus pushes towards the periphery opposite to the axon of Hila. That means this nucleus will be present here. This nucleus will be pushed towards the periphery. Right? So these are the changes which occur in the cell body. So within 48 hours after injury, this chromatolysis will happen. Right? Then what are the changes which occur in the proximal stump? And this is distal stump. No? First we will discuss what are the changes which occur in the proximal stump. In the proximal stump, the changes which occur in the proximal stump, what we call retrograde degenerative changes. What are those? Retrograde degenerative changes. So changes which occur here, these changes what we call retrograde degenerative changes. So what are those? Very simple. Fragmentation of axon cylinder, breakdown of myelin sheath. Up to where? Up to next node of Ranvier. That means up to here only these changes will occur. Right? Where? In proximal stump. Then changes which occur at the site of injury. So what are the changes which occur here? Here, proliferation of squam cells and bridges the gap between two stumps up to 3 centimeters. If it is 3 centimeters, squam cells can bridge. If it is more than this, we have to suture both stumps. Clear? Then, after that, what are the changes which occur in the distal stump? In the distal stump, changes what we call Wallerian degeneration. This Wallerian degeneration we can also call as antegrade degeneration. Right? So now we will see what are the changes which occur in the antegrade degeneration. Fragmentation of axon cylinder. Fragmentation of myelin sheath into small fatty droplets. Right? When these changes occurring, simultaneously there will be multiplication of square cells. These square cells will eat up. These square cells will eat up. That means these square cells will engulf these debris. What are these debris? Fatty droplets and fragments of axon. So they will eat up, or they will engulf. Once they engulf, this total axonal tube filled with proliferated or newly divided squam cells. So now squam cells are present here. Right? Of course, these squam cells contain nucleus. At this time, it appears as nucleated cellular band. See this? It is looking like nucleated cellular band. This nucleated cellular band, what we call band of Bungner, right? So this is band. Next, what will happen? This stump is there, no? Proximal stump. The axon cylinder of this proximal stump will undergo swelling, right? So with this, degeneration completed. Now regeneration will start. How? I already told you, when it's undergoes swelling, from this swelling or from this dilatation, there will be origin of 8 to 10 axonal sprouts. One of these axonal sprout grows and unites with distal stump, and the communication between the proximal stump and distal stump has been established, and the surrounding cells which are present around this axon cylinder, that means these squam cells forms myelin sheath, and the remaining axonal sprouts are there, no? These remaining axonal sprouts disappear, right? So this is regeneration. Very simple. Enlargement at the proximal stump gives 8 to 10 axonal sprouts. One of the axonal sprouts unites with distal stump, right? So that communication established, and the cells which are present around this axonal cylinder forms myelin sheath. That means squam cell forms myelin sheath, right? Here, this squam cell forms myelin sheath, and the remaining axonal sprouts are there. Only one will come and unite, remaining will degenerate. So again, in this cell body also, all the changes which occur in the chromatolysis will reverse, right? Now regeneration completed, right? So this is about introduction to neuroanatomy. Next class we will discuss about cerebrum, right?